Hello, everyone, and welcome. In Chapter 2, we discussed how to view the invisible world, and in this chapter, we'll focus on the different components and functions of a cell and their morphology. The learning objectives for this chapter will be to compare prokaryotes to eukaryotes, learn about different morphology and cell arrangements, understand the effect of osmosis on cells, learn about different parts of the cell and their function, analyze the fluid mosaic model, understand how molecules move across the membrane, identify the parts of a eukaryotic cell and their roles, and define the endosymbiotic theory and the steps involved. We'll begin by classifying cells into two main types, prokaryotic and eukaryotic. You may recall from earlier biology courses that prokaryotic cells are smaller and simpler than eukaryotic cells, with the key distinction being that prokaryotic cells lack a nucleus. A nucleus, an organelle, shown in this image, is a membrane-bound structure that houses the cell's genetic material or chromosomes. The term prokaryotic and eukaryotic reflect this distinction. Pro means before and eu means true, referring to the presence or absence of a nucleus. Microbial cells can belong to either category, or one or the other category, while viruses are neither prokaryotic nor eukaryotic and are not considered cells. We'll explore viruses later in the course. This classification is based on how DNA is stored, which profoundly influences how genetic information is used. Prokaryotic cells, the oldest lineage, store DNA in a non-membrane bound region called the nucleoid. In contrast, eukaryotic cells enclose their DNA within a membrane-bound nucleus, highlighted with purple dashes in this image. If you need a refresher on cell membranes, their composition and function, now is a good time to review. Examples of eukaryotes include humans, while prokaryotes include bacteria such as E. coli. However, not all eukaryotes are multicellular, and single-celled microbes like yeast are also eukaryotic. Here we list a few major differences in the way prokaryotes and eukaryotes conduct things. Prokaryotes and eukaryotes can be divided amongst other traits as well, of course, but where the DNA is located is the most obvious. Prokaryotes contain a nucleoid, which means nucleus-like, and it is an irregularly shaped region within the cell of a prokaryote that contains all or most of the genetic material. The core genetic material of a cell is called their genome. Chromosomes are another name for the structural organization of an organism's DNA. In prokaryotes, their chromosomes are circular, meaning they have no ends. However, eukaryotes are not circular. They come in linear segments that have a beginning and an end, and they are not bound to each other in a complete circle. An organelle is a compartment inside the cell that serves a function that is surrounded by a membrane. The first example of this we've discussed so far is the nucleus, but eukaryotes have many of these organelles, while prokaryotes don't really have them. There are some unique exceptions, but in general, eukaryotes have many organelles, and prokaryotes you should think of as not having organelles. Prokaryotes have a strong material in their cell walls, and this is called peptidoglycan, that lends to the strength, structure, and shape of the cell. Depending on the eukaryote, they may not have any extra cell wall material, but some eukaryotes instead have chitin, cellulose, or other materials than peptidoglycan that we won't get into. DNA packaging is extremely important. Depending on the organism, DNA can range from millions to even billions of individual units bound together that make up the genetic code. The cell needs a way to package that information neatly. Eukaryotes have proteins scientists call histones that wrap up, pack, and package the DNA. Looking at this image from right to left, we can see the unpackaged DNA. Histones are used to wind up the DNA into coils, and then those coils are coiled again upon themselves to form what are called supercoils, which are the structure that is condensed to form the linear chromosomes of eukaryotic DNA. Finally, prokaryotic replication occurs through binary fission, whereas in eukaryotes it occurs through mitosis, or a process called budding. We will cover budding in more detail in Chapter 5, and mitosis was already thoroughly covered in Bio 181. Recall that there are three domains of life: the eukarya, archaea, and bacteria. We will focus primarily on bacteria because they are well studied and they are responsible for a significant amount and types of human diseases. Also, because we know incredibly little about archaea and their roles with humans. We will also briefly cover eukarya, which contain the microbes, fungi, protozoa, algae, and helminths. And when we later cover acellular microbes, we'll get into greater detail on viruses, but this chapter is about the cell. So let's dig into bacteria and prokaryotes a little bit more. Morphology describes the form of the cell. Take a moment and try and remember why cell morphology. Take a moment and try and think about why cell morphology is so important to microbiology. If you said it's important because it's necessary to identify microbes, then you're on the right track. Prokaryotic cell morphology comes in three different shapes. Coccus, from the Greek meaning round, spherical, or ovoid. Bacillus, which is a rod-like shape or pill shape. And spiral, which is corkscrew in shape. Note in this image, the coccus, which is on the left-hand side, can sometimes have the appearance of a bacillus bacterium. And that is due to the fact that this is a particular bacterium that is going through binary fission. And if one looks carefully, they will see a little indent in the center of that pill-shaped pair of bacterium in the cell wall. And that actually indicates that this cell is going through division, and those are the two cells in the process of forming and separating. If the word ends, and just as a side note here, as we're getting into these different scientific terms, if a word ends in us, us, it is the singular form of that word. So, for example, bacillus is a rod-like bacteria in shape, and that is talking about a single bacterium with that shape. If the word ends in I, it is the plural form. So if we're talking about bacilli, we're talking about a group of bacteria that are pill-like in shape. Bacteria also have different arrangements. In this image, you can see diplococci and streptococci. And what determines the arrangements is the plane of division that occurs in the bacteria. The plane of division is along what axis the cell breaks apart into two new cells, shown here as a blue square that intersects the cells. If a bacterium divides along the same plane for each of its progeny, and they do not separate, then you can either have diplococci, meaning two coccus bacterium, or streptococci, meaning a chain of coccus bacterium. Diplococci is when they only stick together in pairs, therefore, and streptococci is when they stick together in long chains. Note the size of the bacteria we're looking at with a scale bar. These microbes are in the range of about 0.8 to 1.5 microns in size, which is very typical for bacteria. Streptococci can be miscategorized by students as spiral bacteria because they can make what appears to be a spiral shape when you link many of them together, but you need to look carefully and see that they are made up of individual cells. Because plane of division determines the arrangement of the bacterial cells, if a bacterium divides on two different planes that intersect each other at 90 degrees or at right angles, as in image B, this division pattern leads to a tetrad, meaning that there are 4 bacterial cells that can remain as a group. If a bacterium divides on 3 different planes, and sticks together as a group, or of 8, we can have a sarcinae, as shown in image C. Lastly, if we have a bacterium that divides without a set plane, then we can have clusters that look like a bunch of grapes, and that is called staphylococci. Most bacterium separate after division. In this example, and what I mean by that is into two different cells that are no longer linked together. In this example here, we have a single bacillus, and occasionally you might see single bacillus attached to each other. But as we can see, these cells are not a full-length cell. So this indicates that these cells are actually still going through binary fission. However, in example B, we can see that these cells can often remain as partners, even as we see here in their full size. So these are referred to as diplobacilli. If you see in a culture that the majority of cells are diplobacilli, and they are for the most part, they look like they're full-grown cells, you are likely looking at a true diplobacilli. If you see many of them as single bacterium, and the ones that are still attached look like they're smaller versions of the single bacterium, you're probably just looking at single bacilli. If these bacilli remain attached by their polar regions, in other words, their ends, say if you're looking at a pill-shaped or a bacillus form of bacterium, then we have streptobacilli. Sometimes these bacterium are somewhere in between and have an identity crisis, so to speak. So these bacterium are called coccobacillus, shown in image D. They are truncated forms of true bacilli, so they're not quite as long as what you see in the streptobacilli of C. And again, they are not quite as completely round as you would see with a coccus-shaped bacterium. So they're not quite as slender as a bacilli, but not quite as round as a coccus, and that gives you a combination of both coccobacillus. Not all bacterium choose to be perfectly straight, and some bacterium have curves in them. If a bacterium has a single curve in it or a mild curve, it is referred to as a vibrio. If that bacterium has many curves in it and has this rigid corkscrew-like morphology, it is called a spirillum. I want you to notice here with the spirillum the thickness of the wall in relationship to the length of it. Spirillum are relatively thick and spiral-shaped. On the other hand, this is not to be confused with the spirochete, which is thin and long. Because of this, they are springy and they have a different sort of action to the cells. Carefully compare this to the stubby form of spiral bacteria in the prior image, which are much stiffer in design. Okay, before we discuss the cell wall, we need to review one of the most important functions, the movement of chemicals in and out of the cell through the cell wall. Osmosis, or I should say cell envelope. Osmosis is the movement of water in and out of a semi-permeable membrane in order to balance out the chemical concentrations. By definition, a cell has a membrane that surrounds it. This membrane does not prevent water from moving in and out and is therefore referred to as semi-permeable. Water is too small to prevent its movement in and out of the cell membrane. If chemicals like salts are really high in concentration, in other words, the number of chemicals per volume of water is high outside versus inside the cell, water will move either in or out to try and balance out the chemical concentration. In biology, water has chemicals dissolved in it. We call such chemicals the solutes, and we call water the solvent. Examples of solvents you may be familiar with besides water include things like acetone and fingernail polish remover, mineral spirits, or ethanol. The most common example of a solute in nature are salts. Put salt in a cup of water. The water will act as a solvent and dissolve the salt, which becomes a solute. In example A, The ratio of solute to water molecules is relatively the same inside and outside of the cell. We call this isotonic. Tonic is the fluid surrounding the cells. Think about tonic water that you would buy at the grocery store. The tonic is the fluid that the cells are floating within. In example B, the solute to water ratio is higher outside of the cell than it is inside of the cell. In other words, there's more solutes on the outside of the cell per given amount of volume. This is called a hypertonic. Hyper means high, so there's a high amount of solutes outside the cell. Therefore, to counteract this imbalance, water rushes out of the cell because the solutes are trapped and they cannot equilibrate or move inside or outside of the cell. This water rushing out of the cell dehydrates the inside of the cell until the concentration reaches the point where the solution becomes isotonic again. Or the cells are destroyed through a process called crenation. As you can see in the image in B, the cell can have these little indents, which can cause damage to the cell wall as it dries out. And that causes something called crenation, where the membrane can tear or cause plasma, or another word for that is plasmolysis, if the cell expands and breaks apart in the reverse process. So this tearing can also happen when too much water enters the cell, and we call that cytolysis or osmotic lysis, and we can see that in example C. In example C, the solution is hypotonic. And hypo is the opposite of hyper. So this means that there's low solute concentration outside of the cell. And so since we cannot move the solutes across the cell envelope, water will rush through the membrane into the cell, diluting the solutes until an isotonic solution is reached, or we have a osmotic lysis of the cell. And here we can see the movement of water in this image. In an isotonic solution, water's movement inside or out of the cell is equal to each other. And this is what we call a homeostasis condition. And the cell's wall is unaffected. In a hypertonic solution, more water exits the cell to try and balance the concentration of salts. And finally, in a hypotonic solution, more water enters the cell to try and dilute the concentration of salts in the cell. All right, let's now discuss the parts of the cell. We've briefly introduced the nucleoid, which is the region containing the bacterial chromosomes, but we also have plasmids, which are what we call extra chromosomal genetic elements. That is to say that they are not a part of the nucleoid, and they are separate or extra little pieces of genetic information, and we'll have more on them later. We can see in this image the nucleoid region. The dotted line outlines where it is. It is not contained within a membrane, but it's kind of a condensed area of DNA found within prokaryotes. This is difficult or impossible to see using a light microscope. However, it can be viewed with a transmission electron microscope. If you're viewing bacteria with an electron microscope, you can see it, but with a light microscope, don't expect to be able to identify one. So if you do see something that looks like it contains genetic information, it might have a membrane around it, that might be a strong clue that you're actually looking at a eukaryote. Okay, so more on plasmids. Plasmids are much smaller than the chromosomes, and they are also always circular. But plasmids are like additions of DNA that help in special conditions, not for the basic biological functions that are relegated to the chromosomes. Examples of special conditions are for survival to antibiotic exposure or resistance to other sort of stresses such as temperature stresses. And you wouldn't find these genes encoding for the production of cell wall or the binary fission or the creation of other products in the cell on the plasmids. You would find them in the chromosomes. And you might recall that from our prior chapter that Carl Woese used the ribosomal gene sequences to build the phylogenetic tree. So what is the ribosome exactly? The ribosomes are the location where proteins are synthesized. Proteins are the tools or machines of the cell, and ribosomes are the machines that make the machines. The cytoplasm is the goo or liquid portion inside the cell that everything floats within, and therefore it's present in all cells. Since each domain, eukarya, bacteria, and archaea, need to make proteins, the ribosomes are found in all domains of life, and they're mostly identified by their size. Learning ribosome sizes helps students understand key differences between prokaryotic and eukaryotic cells, supporting concepts like the endosymbiotic theory, and it is also critical for understanding protein synthesis and antibiotic targeting. Therefore, their size are one of those little factoids that you have to learn and memorize and will come up later in the semester. Instead of their size being measured in grams or meters, it is measured using something called the Svedberg unit. The size affects how fast they settle in a solution. And when placed in a ultracentrifuge, which is a lab machine that spins samples around, this accelerates the rates at which solids settle to the bottom of a tube in a solution. The size of a ribosome affects how quickly it settles, and therefore the settling rate is called a Svedberg unit. Prokaryotic ribosomes that are found in the cytoplasm have a size of 70 Svedbergs, or 70S. Remember that the S stands for Svedberg unit that measures the settling rate, which is dependent on the size and structure. And before we go any further, I just want to point out that for eukaryotic ribosomes, they're a little bit larger. They're about 80 Svedberg units, or 80S. Additionally, organelles such as mitochondria and chloroplasts have their own ribosomes, and they're actually closer to the size of prokaryotic ribosomes, and we'll discuss why that might be the case later on in this lecture. So here is a table just to summarize the overall sizes of the different ribosomes. And archaea, as you can see, are, even though they're more closely related evolutionarily speaking to eukarya, they split a long time ago from us on the phylogenetic tree. Because of that, they still share some similarities with prokaryotes, like having a similar ribosome size. As you can see, they also have an overall size of 70S. So make sure you memorize these values. Inclusions are nutrient storage centers for the cell. Nutrients like glycogen, which are a collection of carbohydrates, starch, sulfur, phosphates, etc., are stored in inclusion, what we call the bodies, inclusion bodies, and they can be stored and used for a time when they're needed. Okay, now that we've talked a little bit about some of the intracellular components of prokaryotes, let's turn our attention to the cell surface. We'll start on the outside first. Some bacteria make a substance made out of different types of sugars. Not really the tasty varieties we all love and know, but there are a variety of different sugars in biology, and it has more to do with the chemical ingredients, carbon, hydrogen, and oxygen, and the ratio that they occur in. Notice glyco sounds like the sugar glucose. So what we have here is a glycocalyx, and this is a sticky layer that exists on as a coat, which is what calyx means, on the exterior of the cell wall. And if you haven't already, I again implore you to take some time to review the biomolecules of life, sugars, lipids, nucleic acids, and amino acids, as you should be well familiar with them for this course. When the glycocalyx is well organized and tightly attached to the cell wall, It is more specifically called a capsule. The glycocalyx or capsule provides shelter in a way for the bacteria against different environmental conditions or from being attacked by others, such as immune cells. In this way, it helps protect the cell from the host immune system and contributes to the cell's ability to be virulent. And virulence means the ability to cause disease. This is why the capsule stain is so important when identifying pathogenic bacteria. For example, as you can see in this image, only encapsulated Streptococcus pneumoniae, those that are in plate A, are capable of causing pneumonia. An unorganized and loosely attached glycocalyx is endearingly called a slime layer. Slime layers allow cells to attach to each other, attach to surfaces, and to form what we call biofilms. 99% of bacterial cells in nature exist in the form of biofilms. As you can see in this image to the right, slime layers also can increase the virulence of organisms. For example, biofilms can form within a catheter, which prevents the organisms from being flushed away as they can attach better to the inner portions of that catheter. So it allows them to attach and grow on that inner surface. And this can actually occur in other parts of the human body and therefore can also increase virulence. Now, moving along, we have a long protrusion that comes off of cells, which is referred to as a flagella. And these can be used like propellers. As they spin around, they propel the cell and allow for something called motility or movement. Bacteria can sense when they're getting closer to a chemical source by how concentrated the chemical becomes as they move. This is called a chemical concentration gradient. And chemotaxy is the process of moving away or towards a source based on sensing that chemical concentration gradient. The same can be true for phototaxy. Cells that photosynthesize light, they may want to move towards the light source. As they move, they can tell which direction the light source is from by the change in intensity or concentration of the light. In these four images, we have different examples of flagella. In image A, we have something called peritrichus. Peri means around, so we can find flagellum all the way around the cell. In image B, we have monotrichus. It is called monotrichus not because it's at one end of the cell, but rather because there is a single flagellum at that end of the cell. And because we find it at just one end of the cell, we call it polar. Think about the planet Earth and how there are the polar regions in the north and the south ends. On image C, we have something called lophotrichus. And lophotrichus means that there's a tuft of several flagellum all in one spot. And lastly is amphitrichus, which means that the flagellum are located in two different spots. In addition to flagella, we also have fimbriae, which are short appendages. They're very thin, and they help the cells adhere to other surfaces, which can assist in forming biofilms as well. And here we have pili, which are similar in structure to fimbriae. They're short little appendages, but they can be used for twitching motility, so they kind of twitch a little bit and that can help the cell move along. But they're important for a very special tool, and that's for the transfer of plasmids between different cells. Remember, bacterium reproduce asexually, so they can't share genetic information like sexual organisms can. However, through this method, they're able to transfer genetic information through a plasmid, through a pili, down the center of a pili, to another cell. This transfer of genetic information is called conjugation in bacteria. In this image, we can see a pili connecting two bacterial cells. And this is how a bacterium can transfer plasmid DNA from one cell to another. This occurs within the same generation. So instead of one generation to the next as a result of sexual reproduction, where genes are transferred on to the next offspring, this is what we call horizontal gene transfer because the genes are transferred directly across from both individual organisms. An example of vertical gene transfer would be how humans transfer genetic information through generation of offspring. So if you imagine a family tree, the genes are going to be transferred to the offspring, which is vertically on that sheet of paper if you were to draw your family tree. And these two are just exchanging information without creating any new cells with bacterium. This transmission of plasmids is very important to bacteria. As previously discussed, plasmids have non-essential genes that allow them to survive certain stressors. One of these can be genes that allow them to be resistant to antibiotics. So if one bacterium has a resistance to an antibiotic, that gene can quickly spread throughout a population through this method of conjugation. There's no need to memorize this information I have on this slide, but scientists are now using plasmids to fight fire with fire. These artificial plasmids, which are generated in a lab, can be introduced into bacterial populations. And what they do is they outcompete the native plasmids, which might have antibiotic resistance genes in them, which weakens the bacterial population. Bacteria have many layers on their cell envelope. And by the way, the cell envelope is a term that we use to describe all of these layers that build on top of each other around the cytoplasm. And working our way in from the capsule, remember that sugar coating on the outside of the cell. And remember, not all bacteria have a capsule in their cell wall. Let's discuss what the cell wall is. The cell wall is made up of different components, and it rests on top of the plasma membrane, which is the innermost membrane. So you can think of the cell envelope as being made up of the plasma membrane or the inner membrane, and then everything else is counted towards the cell wall, and that's all the other layers that rest on top of the cell membrane. Here's an electron microscope image of what the cell wall looks like. It's labeled CW. It rests on top of the plasma membrane, or PM, and helps maintain the cell morphology, the cell shape. That is an important distinction. The plasma membrane, also called the inner membrane, is not part of the cell wall. The cell wall shields the cell from the external environment, and its rigidity resists the tendency of the inner membrane to want to burst apart in hypotonic solutions. Remember, in hypotonic solutions, there's more solutes inside of the cell, and water wants to make them isotonic. So water will rush into the cell and try and cause it to expand. But when it presses up against that cell wall, the cell wall resists that expansion and helps protect that inner membrane. The cell wall is also what anchors a lot of appendages, including the flagella and other important proteins for cellular function. The cell wall contains peptidoglycan. We've covered this a little bit before already. Peptidoglycan is a complex material and is composed of rows of disaccharides, di meaning pairs of sugars, linked by polypeptides, or small chains of amino acids. Again, disaccharides are made of two sugars, and polypeptides are proteins made of short chains of amino acids. And again, I cannot stress enough, if you haven't yet, to review those four basic biomolecules of life you learned in your prior biology course. So there's the disaccharides, which you can think of as these logs here in this image. and the polypeptides which bridge them together. We will talk a lot about gram-positive and gram-negative cells. They each have cell walls with unique compounds and unique spatial arrangements that you need to memorize. Gram-positive cell walls have much thicker peptidoglycan on the outside of the cell wall, pictured on the top of in the picture, that a purple color on the top there. And they also have two acidic compounds called teichoic acids and lipoteichoic acids that penetrate them. Lipoteichoic acids go all the way through and into the cell membrane or the plasma membrane. The peptidoglycan makes the gram-positive cell walls thick and rigid. Remember that the crystal violet iodine complex in a gram stain is really hard to remove from the cells, and that's why they remain purple after staining and destaining. And this is due to the very thick peptidoglycan layer that they possess. We also have what we call the plasma membrane underneath the cell wall, and the plasma membrane is just a lipid membrane structure. The teichoic and lipoteichoic acids improve the stability of the cell wall, and the teichoic acids are only cell wall deep, whereas lipoteichoic acids penetrate all the way down into the plasma membrane, also known as the inner membrane. Now, both gram-positive and gram-negative plasma membranes have porins, and they are just what they sound like. They're small, they're membrane-bound proteins that act as pores or channels for moving things in or out of the cell membrane. This electron microscope image shows how thick the peptidoglycan layer can be in gram-positive bacteria compared to the very thin cell membrane below it. Gram-negative cell walls have a thinner peptidoglycan layer, usually thinner. Fewer layers built up together here is what makes it thinner. So less of those disaccharides with the peptidoglycan bridging. The peptidoglycan is sandwiched by two membranes, the outer membrane and the inner membrane. The inner membrane, again called the plasma membrane, and gram-positive bacteria have it as well, of course, but they have the gram-negative bacteria. They then have a space that occurs between the outer and plasma membrane, and it is called the periplasm because peri means around again, surrounding, so it surrounds the plasma membrane, or sometimes it's called the periplasmic space. Either will do. It is the region where the peptidoglycan is found in gram-negative bacteria. Because gram-positives lack an outer membrane, they do not have a periplasmic space. So be cautious about that if you have to draw these structures. Note that there are no teichoic or lipoteichoic acids. However, there is another substance called lipopolysaccharides, abbreviated as LPS, that is embedded in the outer membrane, shown as the little hairs protruding from the gram-negative outer membrane. You must be able to memorize the order of the cell wall of gram-negative bacteria and gram-positive bacteria, and you should be able to do it. So, for example, starting from the inside of the cell with the plasma membrane, working your way out to the periplasmic space, the thin peptidoglycan layer residing within it, and then the outer membrane, which has LPS embedded into it. And here again is an electron microscope image of the gram-negative membrane and its cell wall. For reference, again, a bacteria is about 1 micrometer wide. As you see there, 1 to 2 micrometers, somewhere in that range in that text box. A micrometer is equal to 1,000 nanometers, nm. The width of the inner membrane or outer membrane is only about 10 nanometers, or 1/100th the width of the bacteria. So very thin. And You can note that the peptidoglycan layer is typically much thinner than we saw in the gram-positive bacterium. By looking in this image here, if I back up, we can see the gram-positive peptidoglycan layer is incredibly thicker. LPS, or lipopolysaccharide, helps with stabilizing the cell's outer membrane. And by the sound of the name, you should be able to deduce that it's made up of lipids and sugars, lipopolysaccharide. So there are three main components here. There is lipid A, which is a lipid portion, and it embeds itself into the outer membrane, and it does so because of the fact that the membrane is made of lipids, and so the two are both hydrophobic and they can interact with one another. The core and O antigen are the parts that are made up of sugars. And before we get much further, there are many ways of drawing chemical structures or of molecules. And the most common way in biology is the skeletal structure. I think instructors often start using them without taking a step back and showing students how they work. So, a skeletal structure in chemistry is a simplified way to represent a molecule. Only the bonds between carbon atoms are explicitly drawn, and they're shown as lines. Carbon atoms are understood to be present at each line's end or at each corner. Hydrogen atoms attached to the carbons are usually not shown unless they belong to a functional group containing another element like oxygen or nitrogen. However, it is important to remember that each carbon atom will always form a total of 4 bonds. So let's take a look at how this works. Here to the far left is the chemical formula for the hydrocarbon propane. Propane is a 3-carbon hydrocarbon. So you can see here that it has 3 carbons and 8 hydrogens attached to each of them. But it doesn't tell you how those hydrogens are attached. It just lists them here. To the right of that, you can see a different way of drawing this. It's called the Lewis structure. You don't have to memorize the way that's what type of structure that is called. But this here is more explicit. It shows all of the carbons, all of the hydrogens, and all of the covalent bonds these atoms share are illustrated with the drawing of a line. And you will note if you count the lines around every carbon has exactly 4 lines or exactly 4 bonds that it shares with different atoms around it. Next, we have what we call the condensed formula structure. And this is a little bit different than just the molecular formula I showed at the very beginning. Herein, each set of hydrogens are couched according to the carbon they are bound to. So the first carbon has 3 hydrogens. As you can see, it's the green CH3, which matches up with the Lewis structure that was the explicit drawn out version of it all. The second carbon has 2 hydrogens, and the final carbon in the propane chain has again 3 hydrogens. And this finally brings me lastly to the skeletal structure. In this model, if another atom isn't drawn in, organic chemists and biologists know that a carbon is implied to be present at the end of each line or corner. So there's going to be a carbon here, and a carbon here in the propane molecule. Additionally, if we don't see any charges, any positive or negative charges on any of these carbons, we know it's going to have 4 bonds. So this end carbon, we know has 1 bond to another carbon. So where are the 3 other carbons? Well, as I described earlier here, the last bullet here, they're going to be taken up. It's implied to be bound to other hydrogens, unless there's some other important functional group like a hydroxyl, which is OH, or an amine, which is NH3. They're going to be implied that they're all hydrogens. And whether or not you know this, you've seen many of these images. So Now that we know the basic uh, premises for how a skeletal structure works, let's look at the structure of LPS represented in such a way. So here we have LPS again, and we can see that the fatty acid tails here are drawn with just a skeletal structure. So at each end and each corner is going to be a carbon, and it's assumed that to fill out the rest of the bonds for these carbons, there's hydrogens. So this very end point is going to have this very end here is going to have a carbon and then three hydrogens around it, and then bound to this carbon here in the middle, which will have two hydrogens around it, two hydrogens, all the way to the top here. And once we get to this final carbon, because it's not completely bound to hydrogens. You see that the drawing will show that there's an oxygen there and an oxygen there. It'll show if there's a nitrogen there. Anything that's not a hydrogen, it's going to need to draw out. And if it's a functional group with a hydrogen, it will then include the hydrogen. So here's an oxygen bound to an H, that's a hydroxyl group, and it's going to be written out. And so you will come across these skeletal structures throughout the rest of the semester. So it's something good to understand. So finally, let's take a step back here, and here's a side-by-side comparison, which is very useful for studying, I will say. And you can note how the outer membrane shields the peptidoglycan layer in gram-negative cells. Here we see this outer membrane, and the peptidoglycan is protected by that outer membrane. Also, the peptidoglycan layer does not interface with the outside world in gram-negative bacteria, like the gram-positive cells do. See, it lays right on the outside here. This means that peptidoglycan, while important, isn't quite as critical for a gram-negative bacteria as it is for a gram-positive bacteria. And in fact, our earliest antibiotics and many of the common antibiotics we use today. So, for example, penicillin, which we discussed was the first antibiotic that was elucidated. It attacks peptidoglycan. And so, based on what we discussed, you can think to yourself for a moment here, why are gram-negative bacterium less harmed by penicillin, for example? Well, chiefly, this is because the peptidoglycan layer is partially shielded by the outer membrane, but also the peptidoglycan's role in maintaining an intact inner membrane is less essential with the presence of the outer membrane, which can aid in structure. So again, as a warning, you will need to be able to draw these structures. It won't need to look as artistically pleasing by any means, but you should be able to just draw some basic lines and point and say that line there is the inner membrane, that line is the peptidoglycan layer, so on and so forth. Kind of what you see in these images here in this electron microscope. Now, while we are discussing the cell wall, acid-fast cells have yet another chemical called mycolic acids outside of their cell wall. These are waxy substances that help protect and shield the cell, similar to how the glycocalyx works. Waxes are similar to lipids in that they are made up of long chains of carbon and hydrogen and are therefore hydrophobic in nature, and they like to interact with other lipids or other waxes, but not with water. Examples of organisms that contain this are the mycobacterium that cause tuberculosis and leprosy. We've already basically discussed the plasma membrane. The plasma membrane is the membrane that houses the cytoplasm and all of the cell's inner contents. It's a selectively permeable membrane that encloses the internal structures of the cell. So essentially, the plasma membrane acts as the gatekeeper that decides what sort of nutrients or chemicals or products go into the cell or out of the cell, and what sort of toxins or other things are allowed to cross the membrane. Some species of bacteria can turn from normal and active cells, called vegetative cells, into endospores. These endospores are relatively inactive cells. that have turned into a sort of a bomb shelter that is resistant to environmental extremes like high heat and radiation. They do so to survive a harsh condition to emerge later as a vegetative cell and during a better time. This table shows the different traits of each type of cell. Vegetative cells tend to be sensitive to extreme temperatures and radiation. That could be things like UV light, gamma rays, things like that. Whereas endospores are resistant to both. Vegetative cells stain as gram-positive. However, endospores don't absorb gram stains. As a result, you'll need a special endospore staining technique we discussed earlier in the last lecture. Vegetative cells have normal water content, and the enzymes that are inside of them carry out the usual chemical reactions of normal cellular activity. Endospores, on the other hand, are much more dehydrated. The book says they have no metabolic activity, but it's sort of true. It's very minimal to almost none, and that they say that just to help you differentiate between the two. Vegetative cells are capable of active growth and metabolism, while endospores are what we would think of as dormant, and they don't experience growth or um, very active metabolic activity. Additionally, endospores can resist chemical penetration. The process of becoming an endospore is called sporulation, and the process of returning to a vegetative cell is called germination. There are 6 basic steps involved in the transition to become an endospore. First, the DNA is protected by being surrounded with membranes. The precursor to the spore is called a forespore. The forespore builds additional membranes around itself. Then a shell called the cortex forms around the forespore. After that, a final protein coat goes around everything. And then finally, the mature endospore is released from the cell. And we won't get into detail for germination, but it's essentially the process of creating a vegetative cell again. Amazingly, spores can hang around for a very, very long time. This research analyzed what is called the ultrastructure of cells, and that just means the subcellular level structure. So maybe what's inside of the cell. And what they found were intact bacterial spores in the skin of an ancient Egyptian mummy. And they showed that the bacterial spore had characteristic appearances with an intact core, a cortex, a coat, the exosporium, or the outer layer of the endospore coat. By now, you've heard me mention membranes several times, so let's take a closer look at what membranes are. This image shows a cellular membrane, which appears as a kind of sheet made up of interconnected molecules. The way this membrane stays intact is similar to how fats or oils would float on the surface of water. This happens because they're lighter than water, that's why they're floating, but more importantly, they group together because in chemistry, likes attract likes. What we mean by that is, well, I won't dive too deeply into the chemical mechanism since I asked you to review the biomolecules of life, but let's do just a quick recap. There are compounds referred to as hydrophilic compounds. Hydro means water, and philic means to love. These chemicals are attracted to water. A great example is water itself, which tends to stick to other water molecules. On the other hand, hydrophobic compounds, phobic meaning a fear of water, do not interact with water very well. Examples of these are fatty acids, fats, oils, and other hydrocarbons like waxes. Instead, hydrophobic molecules are chemically attracted to each other. If you look at the image on the left, you see a phospholipid. These are the small molecules that make up the membrane sheet, and they have 3 main parts. First, there are the fatty acid tails. Again, you can see the skeletal structure of that. So they are made of implied carbon and hydrogen atoms that are linked together. And these chains, they don't interact well with water because they're chemically neutral, meaning they lack any positive or negative charges. In contrast, at the top of the phospholipid, you'll see a phosphate group. The phosphate group has oxygen atoms attached to the phosphate, and those oxygens have a relatively negative charge to them. This difference is key. So the fatty acid tails, being uncharged, they avoid water, they're hydrophobic, while the phosphate group with its charge is hydrophilic and interacts well with water. As a result, if you were to shake up a bunch of loose phospholipids in water, they would spontaneously arrange themselves into sheets, separating from the water. These sheets, known as membranes in biology, are the foundation of cellular structure. So all of the fatty acid tails would align up to each other in one layer with the phosphates on the other side. And yeah, these sheets, we call them membranes in biology. The other component of the phospholipid is the glycerol backbone. That's this portion right here in the middle. And this is just a simple 3-carbon molecule that connects the fatty acid tails to it and connects the phospholipid to it on the other side. So it is called the fluid mosaic model to describe this membrane structure. It's called fluid because the components can move freely. Each individual phospholipid can move around in that membrane plane. And it's mosaic because it's made up of a variety of molecules. If you were to look at this cross section here, you can see there's a variety of molecules that are inside of it, like a mosaic. And that's basically what bacterial membranes look like. In contrast, archaeal membranes have phospholipids with branched chains attached to them, and sometimes they form a monolayer instead of a bilayer. But in this chapter, we're mainly focused on bacterial cell membranes. And now imagine that this inner membrane of a cell, the phosphate groups are exposed to both since there's 2 layers, the red represents the phosphate groups are going to face inward to the inner part of the cell, and the other side, with the other phospholipids, they're going to face outwards towards that extracellular milieu, or the solution, the aqueous solution that they're in. Cells typically exist in aqueous environments such as the ocean or within the fluids of our bodies. And even cells on dry land encounter moisture such as rain or humidity, which is essential for their survival. So you see it in a lipid bilayer, 2 layers of phospholipids. So let's move on to talk about proteins now, proteins associated with membranes. There are 2 main types, peripheral membrane proteins and integral membrane proteins. Remember, peri means around, so peripheral membrane proteins are located on the inner or outer surfaces of the membrane, but they're not embedded within it. These proteins play an important role in processes such as passing molecules along the membrane, which we'll discuss when we explore ATP synthesis and the electrochemical gradient in metabolism. On the other hand, integral membrane proteins are embedded within the membrane. These proteins can be further divided into 2 categories: those that are embedded on only one side of the phospholipid bilayer, and those that span across both layers of the bilayer, known as transmembrane proteins. The membrane serves as both a protective barrier for the cell and a gateway that regulates what enters and exits. And transmembrane proteins act as channels, allowing specific molecules to move in and out of the cell, ensuring that the cell maintains its internal environment. There are a few other ways that transport can be done. One is simple diffusion, and that is the same principle as osmosis. Simple diffusion is when molecules that are small enough that they can just pass through the membrane without any sort of help from proteins. You can imagine water molecules can do this, but other small molecules like carbon dioxide can do this as well. And if you can remember why a net amount of molecules may pass through a membrane, that's because there's a higher concentration on one side versus the other. And those molecules want to move to the other side to balance out the concentration on both sides. That's called homeostasis. So that is simple diffusion. which should not be confused with passive transport. Passive transport still relies on there being a misbalance of chemical concentrations on either side of the membrane. That's called a chemical or ion gradient. But in passive transport, these molecules can't slip through the membrane on their own, so they need a transmembrane protein. Charged molecules and larger molecules often need help crossing the membrane by way of these protein channels. Here you can see the movement of chemicals is facilitated by the protein, and this is called passive transport because it doesn't require any energy input from the cell. This is active transport because the chemicals are being pumped against the concentration gradient, unlike in passive transport or simple diffusion. Look to the right and you'll see a bar showing that there is more sodium, Na+, outside the cell than inside the cell. So if the cell wants to reduce its internal sodium levels by pumping sodium outside, it must expend energy to pump sodium to the external environment, which already has a higher sodium concentration. And remember, through diffusion, chemicals don't want to go from a low concentration to a high concentration. They want to make homeostasis. They want to have an even amount, both inside and outside. So the main type of readily usable energy stored in cells is called ATP. And by breaking a phosphate from the ATP, it provides the energy required to push sodium out against the concentration gradient. This process allows the cell to transport a wide range of different chemical compounds. Okay, so far we have mainly focused on bacteria more than archaea, as they are more numerous and cause disease. Let's now shift our focus to the eukaryotes, highlighted above here in yellow, which includes fungi, protozoa, algae, and helminths. In addition to our table here, you should also be aware that eukaryotic microbes are larger and more complex than prokaryotic microbes. Think back to our scaling chart where protists were a lot larger than bacteria. This is the basic structure of a eukaryotic cell. As we'll talk about later, you see a lot of these different types of membranes that exist inside of the cell. These membranes allow it to compartmentalize different processes and tasks. Like bacteria, eukaryotes may possess flagella, but usually just as long singular appendages. They also have short numerous ones, appendages called cilia, and both of these can be used for propulsion. So here we can see the little cilia coming off compared to these longer flagellum. As a side note, do not confuse cilia with another appendage called fimbriae, which are found in bacteria and used for attachment. In summary, prokaryotes do not have cilia. However, both eukaryotes and prokaryotes have flagella, which can differ in their size, the chemical composition, and also the way they move and help with motility. There are many organelles and intracellular structures that set eukaryotes apart from prokaryotes. But let's again start from the outside like we did with bacteria. Plant cell walls are made of cellulose, and that gives them a very rigid structure. Under the microscope, you will observe them most often in the shape of a square, and that's due to the cellulose, which provides a lot of rigidity. Fungal cell walls are made of chitin. Animal cells and protozoa lack a cell wall. So they do still have membranes, of course, they are a cell, but they don't have any of these other components that would make up a cell wall. On the left, you see the chemical structure of cellulose, a key component of plant cell walls. Cellulose is a polysaccharide made of long chains of glucose molecules linked together in a specific way. This structural arrangement gives cellulose its strength and rigidity, making it essential for providing support to plant cells. Chitin is a polysaccharide similar to cellulose, but with one key difference. Each glucose has a subunit called an acetate, acetyl group here attached. You don't need to memorize these chemical structures. However, understand that this modification makes chitin a strong and, emphasis on the and, flexible material. And their chitin is commonly found in the exoskeletons of arthropods like insects and crustaceans, as well as the cell walls of fungi. On the right, we have a microscopic image of plant cells, likely from an Elodia leaf, which you might be familiar with if you took Bio 181 at CAC. We used it in our microscope labs. The green structure within the cells are chloroplasts, where photosynthesis occurs. And you can see the distinct cell wall surrounding each cell, and that's largely composed of cellulose. And that provides rigidity and keeps the shape of the cells rectangular. There are several structures that both prokaryotes and eukaryotes have or may have depending on the cell. Obviously, this would include the plasma membrane and cytoplasm, without which there is no cell. And some of these elements differ in their structure and size, such as ribosomes. However, an important evolutionary landmark of eukaryotes was developed that enabled spatial separation, regulation, and membrane-related biochemistry to take place. These structures are called the organelles, which are complex membrane-bound structures. There are many organelles and intracellular structures that set eukaryotes apart from prokaryotes. Remember, these organelles are membrane-bound structures that serve unique functions. The membrane helps to compartmentalize certain tasks and also can be involved in unique membrane-associated processes, which we'll talk about more in another chapter. There are two types of let's get into the membrane organelles, and there are two types of endoplasmic reticulum, which are abbreviated as ER. There are rough and smooth endoplasmic reticulums. The endoplasmic reticulum is an organelle made of a network of lipid bilayer. The smooth ER plays a role in lipid synthesis. When cells create something, we call this biosynthesis, meaning created from life. So they play a role in lipid biosynthesis, carbohydrate metabolism, and even detoxification of toxic compounds. The rough ER is studded with membrane-bound proteins called the ribosomes. Can you remember what ribosomes do? They synthesize proteins. And the reason why they call it rough ER is because under the images, if you view a rough endoplasmic reticulum, those ribosomes stud the membrane and make it look rough in texture. The endoplasmic reticulum, again abbreviated ER, are made of a single continuous membrane inside the cell, folded up many times upon itself. The rough endoplasmic reticulum is studded with 80 S ribosomes in eukaryotes, which are the site of protein synthesis. And you can see it here, these in this image here. You see the little dots along the endoplasmic reticulum, and those are the ribosomes. The smooth ER lacks these ribosomes and therefore appears smooth, those membranes. And they have enzymes that associate with them involved in lipid or steroid hormone synthesis. And that's what we need to know about those particular organelles. The next set of organelles to discuss are the lysosomes and peroxisomes, which are two organelles that have a very high enzymatic activity. To lyse means to break apart. Therefore, the lysosome functions to break down chemicals. It contains digestive enzymes, which are biocatalysts that speed up chemical reactions. Digestive enzymes specifically catalyze reactions that break down ingested particles. Lysosomes are also used by cells for housekeeping or recycling damaged or non-functional cell parts, which are sent to the lysosome to be broken down and recycled. peroxisome has a related task to lysosomes. However, it is most notable for its role in fatty acid degradation. So it can break down lipids. It also breaks down amino acids, and it's named for its reactive oxygen species called hydrogen peroxide, which you may have heard before. You may recognize hydrogen peroxide from first aid kits, where its bubbling reaction with wound components is caused by oxygen molecules forming. This process is called oxidation. It's damaging and makes hydrogen peroxide ineffective antiseptic by harming and killing bacteria. Similarly, peroxisomes function as a controlled demolition site within the cell. They use reactive oxygen species, such as hydrogen peroxide, to break down fatty acids and amino acids safely and efficiently, giving the organelle its name. Lysosomes and peroxisomes are a great example of why organelles are so useful. Having specific areas to carry out these tasks allows the cell to avoid uncontrolled reactions throughout the cytoplasm. The Golgi apparatus processes proteins and lipids. Now, it's not breaking down proteins and lipids like we saw with lysosomes and peroxisomes. It's just processing them before they can go on and do further tasks in the cell. Typically, it does this by adding sugar molecules to lipids or proteins to synthesize glycolipids or glycoproteins. This process is called glycosylation, which means the addition of a sugar molecule. These molecules are very important as components of the cell of the plasma membrane and that are used in cell-to-cell communication. The cytoskeleton is the protein scaffolding of the cell. The cytoskeleton is a collection of structural proteins, and these play an important role in transport of intracellular components. They also help in anchoring of components in the cell or attaching the cell onto other things. They also work to contort and contract the cell, which can be important for motility and to help maintain the cell shape. There are three main types that we're going to discuss, and those are microfilaments, intermediate filaments, and microtubules. Microfilaments are the smallest of the three. They are made of two intertwined strands called actin. These are strands of protein, and they are only about 7 nanometers in size, so slightly thinner than a membrane of a cell. You may have heard of myosin, as it's the major protein in muscle tissue and is used for contraction. It can work in concert with myosin for contraction of muscle cells. They are also used for contorting the cell of amoeba, which allows their motility. Amoeba have what we call pseudopodia, which means pseudo means false and podia means foot, so a false feet. And the pseudopodia will stick to a surface. The cell will have microfilaments that move inward and downward. As they do so, this pulls the inner parts of the cell up into the pseudopodia. Let's take a look at what that looks like. So here you can see this microbe using its microfilaments to extend a pseudopodium to grab onto a surface. By contracting those internal microfilaments, the amoeba contorts its cell and forces fluid into the area of the pseudopodium, pulling the rest of the cell forward into that area. And it repeats this process over and over, enabling movement along the surface. So here's a pseudopodium here, and if you look, let me get out my laser pointer. So here's a pseudopodium here, and if you look really carefully, you can see this fluid rushing in and around, and those are those microfilaments contracting and pulling everything in with it into those pseudopodia. Intermediate filaments are slightly larger than microfilaments, about 10 nanometers or about the thickness of a phospholipid bilayer. The intermediate filaments, they're very diverse group involved in cytoskeletal filaments, And they're kind of between the sides of actin and microtubules. So that's why they're called intermediate filaments. We'll discuss microtubules on the next slide. These strands are made up of amino acids, and they play really important roles in cell adhesion. We can see that this cell right here has these filaments that allow the cells to adhere to different substances, as well as they can be used to help position the nucleus in the correct place, hold the right shape of the cell, resist external forces, and anchor to other cells. So it can act as this scaffolding inside of the cell to help internally hold its shape, which is why they often refer to them as cytoskeletal filaments. Okay, microtubules are the last one to discuss here. They are composed of two types of protein called tubulin. It's what you see in this image here. These proteins, they form these hollow tubes that are about 23 nanometers in diameter, and these can be used as a type of framework of the cell. These proteins can rapidly auto-assimilate, like how phospholipids can form membranes and disassemble, and they act as a sort of railway where motor proteins or proteins that can move up and down this railway can be utilized to transport materials across the cell. Here is another image of tubulin. This is the centromere, which you should have learned about when you studied mitosis. And this tubulin, as you can see, just auto-assimilates, as it's kind of being shown here with the tubulin proteins coming together into a polymer. A polymer, poly equals means many, is made up of several monomers, protein units. And tubulin is a polymer that self-polymerizes. In other words, these molecules just naturally bind and form together. They form these long tracks, these long railways. You can see in this image a motor protein that can attach to the microtubule and move along it. You can see it's carrying what is called a vesicle, and a vesicle is just a ball of membrane that contains some components on the inside. The mitotic spindle uses microtubules as well to separate the chromosomes to separate halves of the cell during mitosis and meiosis of eukaryotic cells. When the cell is separating into chromosomes, represented as the red X's, in mitosis or meiosis before cytokinesis, it uses the mitotic spindle to move them into opposite directions of the cell before the cell goes through cytokinesis. Microtubules attach to the centrosomes at the polar ends of the cell and to the chromosomes in order to pull these chromosomes to opposite sides. This way, each cell has its own copy of the chromosomes before cell division occurs. So you can see just what an essential role that these microtubules play in the cell for transporting molecules and separating chromosomes. Briefly, mitosis and meiosis are the ways in which eukaryotic cells divide and reproduce. And this occurs in normal cells in the body. This could be, for mitosis, could be skin cells or bone cells, for example. For meiosis, these are going to be for sexual reproduction. Mitosis is basically an exact copy of one cell into two cells. Meiosis is a step in sexual reproduction, and this yields gametes in the process. So instead of making two of nearly the exact same cells from the original cells, you make gametes instead. And for those of you that don't know, gametes are another term for our sex cells or our ova and sperm. Since bacteria do not perform these cycles and they don't have sexual reproduction, they don't do meiosis. And a little tip to memorize meiosis versus mitosis is meiosis, again, is a step in sexual reproduction, which has the letter E in it, and so does meiosis. So they both share that commonality. I have placed this image here as just a refresher for many of you that can't remember the processes of mitosis and meiosis. This is a great review slide if you want to take some moment to look at it in more detail. In the blue or on the left side, it shows what mitosis looks like. There's one cycle in mitosis resulting in two cells. Each one of those cells has half the amount of chromosomes found in the original cell, whereas in meiosis, there's two cycles here, meiosis 1 and meiosis 2, resulting in 4 cells that are again haploid in nature here. And my apologies, I made one mistake here. In mitosis, the end product actually, the end cells actually have the same number of chromosomes as the original cell, but rather in meiosis, the final cells have half the number of chromosomes as the original cell. Okay, so it's time to switch bases to an organelle that goes through its own division via binary fission. This organelle is called the mitochondria. I'm sure every one of you has heard of mitochondria somewhere before. The mitochondria are the site of what we call aerobic cellular respiration, and the majority of the cell's ATP production occurs. In high school, we all learn the term that it's called the powerhouse of the cell. Well, what does that mean? Why is it called the powerhouse of the cell? It's called that because the main source of usable power to do day-to-day chores around the cell is typically ATP, which you can think of it as just a little, like almost like packing a granola bar or something. So it's very mobile. It can be used around the cell for a variety of different tasks, such as active transport that we discussed earlier in this lecture. Mitochondria make ATP through the process of aerobic cellular respiration, and we're going to talk a lot more about that when we get into cellular metabolism. But for now, let's focus on other components of mitochondria. Here's the structure of mitochondria, and here's an electron microscope image. The first feature to note is that it has two membranes. It has an outer membrane and it has an inner membrane. And this inner membrane has several of what we call cristae. These cristae are folds, also called invaginations, of the inner membrane that helps to maximize the surface area of the inner membrane. If there were none of those invaginations, we can see that there'd be a lot less total surface area to that inner membrane. You may recall that I mentioned that one of the functions of membranes is to conduct membrane chemistry. So the membrane is like the tabletop of a lab, and they're trying to maximize the amount of membrane surface or tabletop so they can conduct more membrane chemistry. And this membrane is precisely where ATP is formed. Interestingly, mitochondria stand out from some of the other organelles we have discussed because their chief function is ATP production, but they also have their own ribosomes. Their own ribosomes are also unique from the ribosomes we discussed earlier of eukaryotes. The typical ribosome in a eukaryote is 80S in size, but the ribosomes in mitochondria are much closer in size to bacterial ones of 70S. They also have an inner membrane and outer membrane that has a space in between, and none of the organelles have two sets of membranes like this. Additionally, they have their own circular DNA that isn't part of the cell's chromosome DNA that we've discussed earlier that is linear in eukaryotes. Only prokaryotes have circular DNA. So this is getting to be a little weird here. So they have two membranes, their own circular DNA, close to 70S ribosomes, and they also make their own energy, their own ATP. So can you postulate where eukaryotes got such an interesting organelle? The chloroplasts are part of a group of organelles referred to as plastids. This is a class of small organelles such as chloroplasts, which exists in the cytoplasm of plant cells, which is another type of eukaryotic cell. They contain pigments that are used in photosynthesis within the cell of algae or plants. Chloroplasts are another interesting organelle. The two makes ATP, but it uses sunlight instead of food sources like the mitochondria in order to produce ATP. Here's a closer look of a chloroplast. It has an outer membrane and it has an inner membrane and an inner membrane space between the two. It has what we call thylakoids, and these thylakoids are another membrane that exists further inside the chloroplast. These thylakoids house the area where photosynthesis takes place, which is another type of membrane chemistry. As you can imagine, they too want to maximize the membranes for photosynthesis, and they do so by how they organize and stack their thylakoids. The stroma, which is like the cytoplasm for chloroplasts. Um, then the thylakoid lumen is the fluid inside of the thylakoid membranes. So we have the stroma and the thylakoid lumen. And the thylakoid lumen is a space inside of the thylakoid membranes. If you remember, to help you remember this, if you can remember that lumen is a measure of how much light a light gives off. So if you go to the store and buy a light bulb, that's one of the things you can look at the label to see how much light that light bulb is going to give off. The more lumens, the more light. So that might help you remember that the thylakoid lumen is the fluid within the thylakoid membranes. And the thylakoid membranes are stacked very tightly into stacks we call granum. So you can see this stack here looks like a stack of chips. That is called a granum, which is a stack of thylakoids. Like mitochondria, chloroplasts have their own DNA and ribosomes and make proteins. So why is this the case? All other organelles relegate the task of storing DNA to the chromosome and making protein to the cell's main ADS ribosomes found on the eukaryotic cell's rough ER. What is going on here? Scientists began hypothesizing a long time ago that these mitochondria and plastids were originally bacterium. These bacterium lived in some sort of close association with an ancient eukaryote-like organism, very closely related to archaea, modern archaea. The bacterium were engulfed, but not destroyed by the host cell, and eventually they co-evolved to have permanent residence inside of them. There are several similarities which are listed here. For example, chloroplasts and mitochondria are about 2 micrometers in size. That's roughly the same size as a bacterium. And more importantly than them having DNA, what is compelling about that fact is the DNA, when sequenced, kind of like how a paternity test is done. Remember phylogenetics from chapter 1. The DNA is most closely related to bacterial DNA. So in a phylogenetic analysis, we would show them somewhere in the bacterial domain. And their ribosomes are about the same size as bacteria, more closely related in structure. They even divide through binary fission, not mitosis, while all the other organelles don't divide at all. They are synthesized from scratch. Here is a depiction of the evolutionary process called endosymbiotic theory. Sym means to live in harmony with something. Endo means to go into, as you might remember with endocytosis, a particular process for taking in outside matter into the cell. This theory states that mitochondria and chloroplasts arose from prokaryotes that were engulfed by a eukaryote-like organism related to archaea and then lived in symbiosis with them until the bacteria lost many of its traits, making it somewhat unrecognizable. And it needed to survive, didn't need those any longer to survive in the internal world of the eukaryotic cell. And it evolved to better serve as an organelle to provide ATP production for the host cell. Plants and algae had a second endosymbiotic event than just the mitochondria being taken up, and that would be the event that led to modern plastids such as chloroplasts. There is a very robust amount of evidence to believe that these events occurred. As we discussed, one is the size, one is the structure and size of the ribosomes, as well as the size of the organelles themselves. Even you can study the proteomics, so studying the proteins in the mitochondria and plastids. They're more related to protein families within bacteria. In case you're still having a hard time digesting endosymbiosis theory, pun intended, and you can't accept that you have little descendants of bacteria within your own highfalutin cells, let's not forget that evolution is a process, not an endpoint. Endosymbiosis theory is not some relic of the past, of course. We see it in mitochondria um, related to bacteria and chloroplasts, but here's just a brief, non-exhaustive list of different sorts of organisms that have these endosymbiotes that live within them more recently. Dr. Geil is a professor at ASU that I've had the pleasure to work with in studying endosymbiosis, and they had this paper they put out a few years ago. Here we can see algae called Pollina chromatophora, which has endosymbiotes. Endosymbiotes are not quite domesticated into permanent organelles. They can be transient or permanent. So just like the endosymbiotic theory states, Pollina chromatophora actually consumes cyanobacteria, which are little photosynthetic bacteria, but doesn't destroy them. The cyanobacteria can live in and out of the host, and there are several other examples listed in this figure that you can take a look at if you'd like about endosymbiosis as well. Interestingly, scientists have recently found a correlation of the size of the DNA in an endosymbiote and the time period in which it occurred. So in this image here, along the x-axis, we're looking at the size of the total amount of DNA or genome size. And this is in mega base pairs. So DNA is it's a double helix. There's a pairing of nucleotides, but this is basically a string of how many letters are in an organism's code. On the y-axis, it's showing the number of protein coding genes. And as I said, throughout time, these organisms lose some of their traits they needed to survive on their own because now they're living inside of another cell. They don't need to hold on to those traits. And what they've noticed is that earlier the endosymbiotic event occurred, so the longer times, the longer it's been since the endosymbiotic event had occurred, the more genes are lost. So we discussed mitochondria was the first, and they have the smallest genome sizes and the fewest number of proteins that they code for. We said that plastids such as chloroplasts came next, and you can see they have a little bit bigger of a genome size and more protein coding genes. And there are these other events too here. Insects have endosymbionts, and there are other bacterial genomes that we can just list that are free living that have even larger ones. So this is a very beautiful correlation here we see. On that note, sap-sucking insects such as mealybugs, they live their entire life on the juices of plants. If you are ever out and see a pear cactus with white cotton-like substances on it around campus, this is caused from a scale insect that feeds on the sap of a cactus. If you were to go up to the white cottony substance and you were to crush those insects that are inside of it, you will see this beautiful crimson color that's produced. And it's actually a dye. That dye is called carmine. And this dye has been used by pre-Hispanic cultures for a very long time. And its production by the insect is related to the endosymbionts that it carries within its cells. See, mealybugs are not capable of deriving all of the nutrients from the juices of plants, such as cacti. And so they have worked out an endosymbiotic deal with bacterium that live inside of their cells and synthesize those nutrients, such as certain amino acids. And so these Endosymbionts are not responsible for producing the ATP of the cell, but rather for amino acids that are essential as a food source for the mealybugs. Now, things are going to get even weirder. Scientists have found that the endosymbiont found within the cells of mealybugs that produces those needed amino acids of the cell cannot survive within mealybugs on their own. In fact, it lacks certain key enzymes in the metabolic pathway of peptidoglycan synthesis. These endosymbionts have therefore gained their own endosymbiont within it that produces these enzymes. So this is a truly bizarre microbial version of Russian nesting dolls. You don't have to memorize what's on this slide. I just find this kind of an interesting piece of information to end this chapter on. As we continue with this course, I recommend that you begin to reflect on who we are as humans, what strange steps that led to the culmination of humans means for who we are, what we are made of at the cellular level, and how you perceive us, and our place in the biological world. So at this point, we've seen that we are more similar to fungi and plants than many bacteria are to other bacteria. Additionally, we've seen that our cells have endosymbionts that are the descendants of bacteria. In the future, we'll see how our genomes have traces of viruses and other transgenic elements that we've picked up from the environment. So I'll leave you with that. Thank you all for your time, and this concludes our coverage of Chapter 3.